Good evening to all IFPH doctor, members, colleagues, and students, and a warm welcome to all our IFPH English session. Today is our 2097th day of uninterrupted daily webinar of IFPH on 29 May 2026 at 8:30 p.m. Before proceeding, let's have 1 minute silence. Today's our guest, Dr. Suraj Singh Bhadoria, sir. He is MD Homeopathic PhD scholar. He is Assistant Professor Department of Homeopathic Pharmacy, Parul Institute of Homeopathy Research, Vadodara, Gujarat. And he made a lot of research before this session. And his today's topic is Advanced Pharmacy in Homeopathy. A very warm welcome, sir, to you. Can you start your
Thank you, madam.
presentation now?
Thank you, ma'am. First of all, good
You're
evening to everyone. And today we are going to discuss about a very important pillar of recent advancement in homeopathy, especially the homeopathic pharmacy, because pharmacy is the mother for all this growth and development of the pathy. So I just started for the sharing of my slide. Just wait. Now I am going to share this. Is it going to visible to all, ma'am?
Yes, visible.
Okay, sir. Thank you so much, sir. Thank you, ma'am. Today our topic is homeopathic pharmacy in the present scenario. So first of all, what is pharmacy? So pharmacy is the branch of science which deals with the complete knowledge of drug. Okay, so this is the branch of science. So complete knowledge of drug in the sense of identification, in the sense of selection, in the sense of collection of drug material, in the sense of analyzing the drug material, in the sense of preservation, in the sense of preparation, standardization, drug proving, which we also called HPT, homeopathic pathogenetic trial, and afterward the various routes of drug administration, dispensing also, which is known as pharmaconomy. So generally for identification we have so many sources. We have animal, we have plant, we have synthetic, we have imponderabilia, we have isotes, we have allersotes. But in the recent advancement we have some newly added sources. One of the newly added sources is tautopathy, which is the synthetic one, isotes, allersotes. So these are the new method or the new sources through which we can identifying the drug substance. So this is one of the mode, because animal, plant, mineral, sarcodes, imponderabilia, these all are the previous one. These are the classical one. But allersotes, isotes, tautopathy, which is also known as a synthetic source, these are the newly added sources in the collection or the identification of drug substances. Now today, when we talk about collection, so there are so many procedure and the rules and regulation given by the pharmacopoeia. When we talk about homeopathic pharmacopoeia of India, so there are certain monographs which are in regards to the particular collection of drug material. So it is in respect of time, it is in respect of the particular climatic condition, it is in respect of a particular zone of the soil. So the collection of drug material, it is in regard with the collection of that active principle, which is the secondary metabolite. Because ultimately in any drug substances there are two kind of metabolite, primary metabolite and the secondary metabolite. The primary metabolites are generally, they are carbohydrate, fat, starch, so they are not essential for the therapeutic action. But here the secondary metabolite, the active principle, they are considered as a main purpose, yeah, or main aspect for the therapeutic action. Suppose in belladonna the atropine is the active principle. Okay, in Rauwolfia serpentina, serpentine is the active principle. So these are the various metabolites. So what we are going to do? We are going to collect the active principle along with the primary metabolites also. So these are important for us. Next is analyzing. So analyzing in terms of quantification, in terms of qualification. So nowadays we have so many tools like Raman spectroscopy, which is discovered and invented by Dr. C.V. Raman, Nobel Prize winner from India. We have XRD, X-ray diffractometer. We have atomic force microscopy, AFM. We have UV visible spectrophotometer. We have HPTLC, GC, HPLC, LCMS, liquid chromatography, mass spectroscopy. So there are certain methods through which we analyze or optimize the major constituent of drug substances. So this is the analyzing. We are going to optimize the basic content of that particular drug substance. So this is the most important part. Analyzing, analysis. Next is the preservation. Here we are going to preserve the again active principle, because active principle are important for the therapeutic action. If they are going to damage, and if there are certain modulation and modification alteration in the active principle, so they may hamper. So these are the preservation. So this is all about the general aspect of pharmacy. So when we talk about drug proving and the drug development, so as per the CCRH guideline, we have phase 0 study. So what is phase 0 study? It is experimental pharmacology. So what is experimental pharmacology? When we go for the testing of efficacy and the toxicity of any drug substance in vitro model or in vivo model, so these are considered as a drug proving, drug development. So these are the new guidelines. These are the homeopathic pharmacy in the recent scenario, in the recent advancement. Because as per the organ on philosophy, materia medica, we generally based on clinical trial, clinical pharmacology. But before entering into clinical pharmacology, we must have experimental pharmacology, which is phase 0 study, in vitro study and in vivo study. So this is the most important part for the recent development in the homeopathic pharmacy. Now, when we talk about in general aspect for the pharmacy, so it is in regard with the production, discovery, preparation part, dispensing, reviewing, monitoring the medication action for the safety, for the efficacy, which we can say ADR. So what is ADR? Adverse drug reaction. So this comes under the pharmacovigilance. So pharmacovigilance is one of the branch of pharmacy. So pharmacovigilance, it deals with the stoppage or refusing of those drug material or the medicinal product which causes side effect, which causes adverse effect. So these are the basic aspect and the objective of the pharmacy in the field of science and the art. Why it is art? Because ultimately we are preparing medicine in a dynamic level by the virtue of potentization. So that's why it is the procedure of art. If there is no potentization, if there is no dynamization, then it is not art. It is a science. So this is the aiming to ensure the safety, efficacy and the adorable use of the medicine. And the most important part is it is in link with the health science, with the pharmaceutical science and the natural sciences. So these all are the pillar of pharmacy: health science, pharmaceutical science, natural sciences. Pharmaceutical science, when we go for preparation part, preparation of external application, preparation of various ointment, medicine, drug, so these are the pharmaceutical sciences. ठीक है? So this is all about the basic aspect of the pharmacy. Now, when we talk about this clinical pharmacy, so generally this clinical pharmacy, it is in regard with the clinical pharmacology. When we are going to test the efficacy of any drug or medicinal substances on the provers, on the living body, on the multicellular organism. So this is the clinical pharmacology. So there are certain phases of clinical pharmacology: phase 1, phase 2, phase 3, phase 4. So these are in general aspect of the pharmacy. Now, when we talk about identification, as we know, in HPI, in the monographs of HBI, HPCUS, LMHI, ECH, there are certain guidelines for identification. But in the recent scenario, we are going to identifying those drug material or the plant species which comes under the hybridization. So this is the recent advancement, because this part is not even though given in any monograph of any pharmacopoeia. That's why our today topic is the present scenario, the recent application, the advanced application, the recent development of homeopathic pharmacy. So this is the synthetic part. So when we go for the hybridization, so what is hybridization? The hybrid plant are created by the cross pollination. Cross pollination of two different plant species or the varieties, which gives a different trait. So this is the general aspect of hybridization. So when we collect the drug material from such hybrid plant, which is cross pollinated by A and B, which gives AB. So this is the one of the advanced technique which will give you a new variety of morphological feature or a new compound in a drug substance. So this is the hybridization. So this is one of the part. Even though it is not given in any textbook, any pharmacy textbook, Mandal and Mandal, DD Banerjee. So this is very, very rare. When we go for the collection of drug material from the hybrid variety, so it is one of the very challenging job. Why? Because ultimately, when we talk about the collection, when we talk about the identification, so ultimately we are going to identifying the secondary metabolite as well as the primary metabolite. But here, due to the cross pollination, there are more chances of different variety, a compounding effect. What is the compounding effect? When A combined with B, when it forms C, so this is the combined compounding effect. And what is combining? When A combined with B and when it form AB, so it is combining. But we want compounding. A plus B, C. So this is the hybridization. So when we go for such plant material, so they will give you a new conclusivity. They will give you a new idea. They will give you a new characteristic symptom in the plant species. So this is one of the method of identification. Next, when we talk about the selection of drug substances, so there are certain pillar and the factors which based on the drug mapping. So what is drug mapping? Number one, molecular mapping of source. Number two, molecular clubbing. Number three, molecular synergistic. Number four, molecular dynamization. So these are the major pillar for the selection of any drug material, because nowadays we generally prepare a medicine in a bulk, like mother tincture, like 30, they are already diluted, 30 and 200 is diluted, ultra diluted. But where is molecular marker? Where is molecular clubbing? Where is a molecular synergistic? If we say Silicea is the chronic of pulsatilla. So this is one of the molecular synergism, synergistic effect. So where it is written in any phrases or any volume in any part of materia medica, no, it is not written. So this is one of the lacuna in the homeopathic materia medica, because molecular synergistic effect will give you the conclusivity of relation between two different medicine. Either they are follow well by, compare to, compare by, or antidote, inimical. So these are molecular synergistic effect. So generally in the molecular marker, we have genetic diversity, we have genomic mapping, we have gene pyramiding, we have hybrid purity, we have parent line identification, we have DNA fingerprinting. DNA fingerprinting, it is not already in regard with the human DNA fingerprinting. For plant species also, because they are the living trait. So for that particular species, for their identification in terms of the life sciences, we go for this type of fingerprinting, which is known as a DNA fingerprinting. Yes or no? Yes. So this DNA fingerprinting will give you an idea about the selection of that particular marker, which is important for the optimization, for isolation. Because ultimately, when we are going to optimize, when we are going to isolate, when we are going to identify such compound, then only you can say yes, they are the specificity in homeopathic pharmacy. But sorry to say, in our pharmacopoeia, in any pharmacopoeia, in any volumes of pharmacopoeia, these things are not written. So these are very, very important and star point. It must be. So when there is a optimization of DNA fingerprinting in their concentration, in their marker, in their what we can say the specific compound morphology, then only we can say yes, our medicine is already standardized. Got it my point? So standardization itself is a huge part. It is one of the very huge part. So that's why we want to make a holistic approach for this selection of drug material. So there are so many pillars. Okay. Now next, when we talk about this particular collection of drug material. So in the previous era, it is not recent advancement. In our Hindu mythology and in our ancient time, when we talk about the collection of drug material, when we talk about the Ayurveda, which is one of the oldest system and the school of medicine in the India. So generally, before collection, we go for the prayer. So this prayer, these are the dynamic influence. The dynamic influence in between the carrier and the plant trait. So they make a certain connection. This is called the quantum mechanics. Okay, so it is one of the method of quantum mechanics, the part of physics. So there are certain vibrational theory, there are certain photon carrier particles which transmitted from one level to the another level. Have you heard about Bohr's atomic theory? So this is one of the method of the collection of drug material. Okay, so before going to collection, we have to go for prayer, we have to go for such type of ethical and not ethical, the religious practices, so that we can make a dynamization, a effect of dynamization in our drug material. So these are the most important part. Again, it is not written in any pharmacopoeia, homeopathic pharmacopoeia, the method of collection of drug material. So that must be included. So this is very, very important part. Every time it is not mandatory, we have some evidence, we have some scientific evidence, we have some literature, and we have some theory, we have some evidences. There are certain phenomena, there are certain hypothetical part which make the artistic approach in our evidence-based medicine. So this art is needed for the methodology, for the collection. So this is from our mythological background. So there are so many history, there are so many stories also, which are resemble with the collection of drug material. And there are so many characters also in our Ramayana and Mahabharata. So they are, before going for the war, they are going to collect, they are going to pray, then they are make a paste, then they are make the herb, then they are going to making the what we can say the route of transmission. So this is already written in our various scriptures and the mythological background. So recently it was in a somewhere missing in our pharmacopoeia, because we are now advanced in terms of science, evidences, not in terms of art. So that must be needed. So this is very, very important for the collection. Next is preservation. When we are going to preservation, So ultimately what we are going to preserve? We are going to preserve the molecular dimension. We are going to preserve the chemical and the physical integrity, the physical property, the chemical property. We are going to preserve the metabolites. Which metabolite? Both primary metabolite as well as the secondary metabolite. We are going to preserve the therapeutic dimension. So these are the basic aspect for the drug preservation. Preservation, it is not related to kept the drug material on the cool dark place, away from the sunlight, away from the strong smelling bottles. No, preservation, these are the certain procedure for making the optimization in our drug molecular dimension, in a drug mapping also. So when there is no modulation in the drug property, in the molecular dimension, so ultimately our drug is preserved. When there are certain changes like change in color, change in odor, change in taste, change in texture, change in viscosity, so ultimately our drug is not preserved. It is not going to preserve. So these are the certain method, these are the certain what we can say, the aspect of the preservation. Because ultimately as per the CCRH guideline, again there are no any specificity for the preservation. So when we talk about preservation, so there is one concept which is called shelf life. So what is shelf life? So shelf life is the expiry of any medicine. Okay, so ultimately in our homeopathic medicine, as we know, our medicines are hydroalcoholic based, especially mother tincture. And when we talk about above the mother tincture, the higher potency, so they are also alcoholic base. So it is already diluted. It is ultra high diluted. So where is preservation? Yes, there is a preservation in terms of H and OH bond. So what is HNOH? This is water memory. As we know, water memory is one of the hypothetical part in the ultra high dilution. There are certain water molecules, water memory, which are going to capture the drug power, drug memory, medicinal memory. Ultimately, in our ultra high dilution, there is no atom molecule, there is no ions, there is no isotopes. These are present in the mother tincture. So ultimately, what we are going to preserve? We are going to preserve the vibrational frequency of H and OH bond. So this is the preservation. Next, now what are the different method of the preparation? So as we know, we have three different scale: decimal scale, centesimal scale, 50 millisimal scale. But now we can go for the fractional dosage also. So what is fractional dose? So ultimately these are the basic pillar: baseline sample, standard sample, trial sample, and error sample. So this is a baseline sample. So what is baseline sample? So baseline sample is generally from 1 to 10. So suppose if we make the extract or the what we can say the mother tincture in a different drug and vehicle proportion from 1 gem 1 to 1 gem 2 to 3 4 5 6 7 8 9 10. So these are the baseline sample. So again, it is not written in any volumes and any monographs of any pharmacopoeia, either international or national, HPI. So this is the baseline sample, because we must prevent the bias. Okay, so we don't know about the efficacy of any drug substance in any particular proportion. So what we can do, we have to prepare a different proportion: 1 gem 1, 1 gem 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. So this is the baseline sample. Okay, so one is the drug substance and remaining part is the vehicle. Vehicle may be the distilled water, vehicle may be the absolute alcohol, strong alcohol. Okay, so this is the baseline sample. For external application, it may depend. Suppose if we prepare ophthalmic suspension, so the vehicle may be distilled water, vehicle may be the isotonic solution, which is sodium chloride, and vehicle may be the glycerin, because there are different types of ophthalmic suspension. If we prepare lotion, so the vehicle, the base part of lotion is distilled water. So the main aspect of vehicle is distilled water. So you can change the proportion of this distilled water in a different ratio. 1 gem 1, 1 gem 2, 3, 4, 5, 6. Now this is the base. That's why it is called the baseline sample. It will give you the origin of other main, what we can say, the sample origin or the basic preparation part. So this is the base, this is the ground pillar of the preparation. Now the next one is standard sample. Suppose if 1 gem 3, 1 gem 4 and 1 gem 6 and 1 gem 9, from this 1 gem 1 to 1 gem 10, so they are showcasing more efficacy in both phase 0 study or phase 1. So what is phase 0? That is experimental pharmacology. What is phase 1? That is clinical pharmacology. So if the drug testing or the efficacy of any drug material is more in 1 gem 3, 4, 5, 6, 1 gem 3, 4, 6 and 9. So these are called the standard sample, because now they are going for the trial, further more studies. So this is the phase 0 study. It is called the experimental trial. Got it my point? So this is experimental trial. Now afterward, when we go for this standard sample, it will give you the conclusivity of specificity in a specific drug vehicle proportion. That's why this specificity in a specific vehicle in a specific drug proportion is known as a standard sample. Okay, now the next one is trial sample. Now similar as this standard sample, when they are going to apply for clinical one or post clinical, so this is called trial samples or repeated clinical pharmacology. So there is one phase where we can go for the repeat clinical pharmacological analysis, repeat analysis. So this is called the trial samples. So this is HPT, homeopathic pathogenetic trial. So this is 1 gem 3, 1 gem 4, 1 gem 6 and 1 gem 9, as similar as standard sample. But these are different from the baseline sample. So this is one of the recent advancement in the homeopathic pharmacy. Now next is potentization. Now what are the recent advancement in the potentization? So ultimately what is potentization? So in UG level we can say it is the method of mathematico-mechanical logistic process to enhance the dynamic power of medicine or the drug substance. Correct? For PG, now the definition change. It is the mathematico-mechanical process to convert the mass particle into massless particle which resemble with the certain isotopic molecules and water memory along with the electrostatic or electromagnetic force of attraction. So this is the explanation, the scientific explanation of potentization. Now here in image you can see this is the laser beam application, inclusivity of laser beam. So number one, influence under the beam of light. Okay. Second, space therapy. Third, synergistic inclusivity. Okay, so this is very important for the recent advancement, because nowadays potentization, it is not like a very unique, very different, but the basic characterization of potentization is the conversion of mass to massless particle with the certain water memory and the silicates, silicons. There is one hypothesis which is called silicon hypothesis. So this is the general aspect of potentization. So when we go for this influence of light under this potentization, so this beam of light, it will accelerate the mass particle to massless particle, reducing their weight, reducing their atomic percentage, reducing their molecular percentage by releasing the energy. Got it my point? So there are two reactions, endothermic, exothermic. So when the conversion of particle from mass to massless, so they will always release the energy. There is a lysis, there is a breakdown. Got it my point? And when the two different particle, when massless become mass particle, so they will gain the energy. So this is the basic aspect of the molecular sciences. This comes under molecular sciences. So this is one of the influence of beam light. Space therapy. Now in ISRO, previously we have NASA, but in India we have ISRO. So there are certain projects which is called EMR, extramural research. So they will allow us certain projects from outside, from multidisciplinary, from different sciences also. So they will give the felicitation of homeopathy also for giving certain trials or the different method of potentization and the efficacy or the in vitro study or the phase 0 study in space also. Okay? So this is one of the recent advancement. We can also go for that potentization in space so that we can know the zero value of this particular massless particle, because in space the weight is zero. So this space therapy is the inclusivity of that particular potentization. Now synergistic inclusivity. What is synergistic inclusivity? When this massless particle showcasing some new structure, some new action. So this is the synergism. This is the synergistic effect. So these are the basic, what we can say, advancement, because potentization is not now recently advanced technique. So these are the recent objectives and the advancement in the field of potentization. Now the next is standardization. Now in the recent advancement, just wait. Yes, sorry for this disturbance from the noise. So in standardization, this standardization generally they are the basic parameter to quantify and qualify the drug material. So in standardization, the number one is organoleptic evaluation, which is from color, odor, taste, texture, factors. Number two is botanical. Again, it is very common. There is no recent advancement. Microscopic, macroscopic to quantify and qualify the structure. But in microscopic evaluation, when we go for SEM and TEM analysis, what is SEM? Scanning electron microscope and transmission electron microscope. So when we go for such type of microscopic study, then only you can say, yes, we optimize the basic morphological feature of that particular compound in a particular drug substance in a particular concentration. So this is the recent advancement. It is the novelty, because ultimately we are not in today's scenario in any pharmacopoeia. There is no specific proportion and the morphological feature of any drug material. But this SEM and TEM analysis, they will give you the specificity in the microscopic evaluation. Gotten my point? Next is physical. The physical is the moisture estimation, extract value, ash value, fluorescence analysis. So this is the physical analysis. In chemical one, we can go for quantification, qualification, chromatography, HPTLC, GLC, HPLC, because these all are regarding for the determination of particular chemical ingredient or a chemical compound in a drug substance. So this is one of the chemical method of quantification and qualification of not only drug substance, qualification and quantification of a specific compound and the molecular marker which is present in our drug material. Got my point? The next part is the biological evaluation. The biological evaluation generally denotes with the microbial contamination, microbial testing. So what is microbial contamination and microbial testing? This comes under the phase 0 study, which is called experimental pharmacology. So this is the experimental pharmacology. So here we are going to showcase the efficacy of any particular drug material, any particular formulary, any particular external application, any particular remedy, any particular extract, any particular herb, any particular medicine in a specific microbe. Either they are gram positive, gram negative, or fungal species. So this is the microbial contamination. Microbial testing, gram positive, gram negative, total viable aerobic counts. So these all are in regard with the microbial contamination, microbial testing. So these are in vitro study. Got it my point? Now when we talk about the in vivo model or ex vivo model, there are two models, in vivo and ex vivo, EX, in vivo. So in vivo and ex vivo, these are the testing of the efficacy of any drug material, any medicinal product in the lower animals, whole living body, rat, mice, rabbits. So these are the in vivo, ex vivo. So this is the biological evaluation. So this may plays a major role for the new drug development, for the drug proving, for the determination of the specificity in the specific mechanism, the mode of action of that particular drug substances. So this is the biological evaluation. This is the biological method through which we can make standardization of any herbal drug substance, any medicinal substance, any remedy, any external application. So these are the major pillar. These are the major aspect for the standardization. Quality control. Now moving towards next. Now this is the drug development. So now in the process of drug development, number one, discovery. Discovery of what? Discovery of new source, as similar as isotes, allersotes, synthetic and tautopathy. Discovery of a new compound. Discovery of a new method of the mechanism of the action of the drug material in that particular living entity. Discovery of any particular optimization, discovery of any particular specific concentration or a specific part of the drug compound. So this is the drug development. Next is preclinical research. So this preclinical research is phase 0 study, which is experimental pharmacology. So in experimental pharmacology, we have in vitro, we have in vivo, we have ex vivo. So these are the experimental preclinical research study. Clinical development, now there are certain phases, phase 1, phase 2, phase 3, phase 4. What is phase 1? Phase 1 is the determination of the mode of action of drug or any medicine on the patient. So this is the clinical development. So this is called pharmacodynamic, what the drug does with the body. So this is the clinical development. Now what is pharmacokinetic? So pharmacokinetic, it is again the branch of pharmacology, pharmacy, which deals with the reverse action. It means what the body does with the drug. So this is the clinical development. So phase 1 is action, phase 2 is dosage determination, phase 3 safety and efficacy, ADR, adverse drug reaction, and phase 4, last phase 4, it relates with the post marketing surveillance. So these are the various phases in the clinical development. Now the next is regulatory review. In the regulatory review, we are going to reviewing the all the rules and regulation, all the ADRs, all the side effects, all the adverse effects, all the action, all the mode of action, different pathway of action of any drug or medicinal substance. So these are in our pharmacopoeia. These are in our specific booklet. So this is the regulatory review. The post-marketing surveillance. Now this is the pharmacovigilance. So what is pharmacovigilance? It is the branch of pharmacy which deals with the method of stoppage or refusing the any adverse event or adverse drug reaction of any remedy, medicine or drug material. So there are so many procedures in pharmacovigilance. First we have to identify then select, then we have to go for analysis, then we can go for the trapping of that particular adverse event in PHC center, in hospitalization. Then we can go for the making method to stop that particular usage of drug or medicinal substance. So this is all about the drug development process in the recent advancement, in the recent development in pharmacy. So here we are not only talking about homeopathic pharmacy. Homeopathic pharmacy is after pharmacy. So first we have to know about what is pharmacy in general. Okay, so pharmacy is a very important, and it is just like a sea. It is the huge part. Okay, so this is very important to know the discovery, to know the level of development. There are different level of development: level 1, 2, 3, 4, 5, 6. So in level 1 you can go for the discovery of any particular source. In level 2 you can go for the discovery of any particular compound from that particular drug. In level 3 you can go for the optimization of that particular compound. In level 4 you can go for the optimization in terms of their concentration, in terms of dosage, and an optimal. In last, the level 5, it is in regard with the identifying their action. So where we can identify their action from the literature part, from their taxonomical classification. Okay, from the historical plot and the origin, you can know about the specificity in their mode of action. So this is the various level of development. So this comes under the recent development and the advancement in the homeopathic pharmacy and pharmacy in general also. Now when we talk about this animal trial, so this is one of my experimentation for my PhD. In my PhD, my whole research work is based on first making the specific formulary, which is external application. Then we can go for the optimization of that particular drug material from the standardization, UV visible, HPTLC, FTIR, SAM analysis, standardization. Then in vitro model, microbial testing. Now this is the animal trial. This is the rat model. Okay. So this is my PhD research work on in vivo study. So it is not ex vivo, it is in vivo. Okay. So it is under the whole living body. So this is one of the animal trial. It is phase 0 study. It is experimental pharmacology. So this is the most important part for the any research work. Now this is the emergency handling. In certain cases for animal trial, there are certain ADR, adverse drug reaction. So sometimes we don't know about the specificity in a particular marker, in a particular concentration, in a particular doses. So it may happen because ultimately in pharmacy, homeopathic pharmacy, posology is question mark. Okay, so there is no specific dosage on a specific disease condition. So for this emergency handling, this is a case where we can go for the administration of arsenic album 200 oral doses for this rat model. So this is one of our emergency. So there are certain procedure which are important and which are to be needed for any experimental pharmacology and any phase 0 study. So this is in vitro study. So in this in vitro study, we are going to make a development of culture media. So this is the homeopathic medicated culture media. So this is one of the novelty. This is one of the challenges in the recent advancement, because now there is no such specific homeopathic culture media. So this homeopathic culture media, it is made by the Echinacea mother tincture. So as we know, Echinacea is antiseptic, antiviral, antifungal, antibiotic. But on other hand, this Echinacea compound also helpful for the proliferation of the colonization and the growth of that particular microbe or the bacilli or the microorganism. So this is bipolar. Every particle denotes in a biphasic nature, plus and minus, dextro and levo. So similarly, in this in vitro study, we introduce a new method of development of homeopathic culture media. So what is homeopathic medicated culture media? It is the culture media which is made by the mixing of homeopathic medicine, which is the tincture, which is used for the growth of various microorganisms. Either they are gram positive, either they are gram negative, either they are fungal variety. So you can see this is the couplet method. This is the classical method. In pathology, microbiology, this is one of the branch. This is one of the procedure through which you can know about the toxicity analysis through or the efficacy of any drug material, or you can also make a optimization. So this is new drug development in the field of microbiology, which is homeopathic medicated culture media. So this is in correspond with the pharmacy college. Okay, so this is in vitro study. Now, this is the research work interdepartmental. Again, this work is already published. So this is the showcasing inhibition zone of homeopathic culture media in fungus and bacterial, both fungus and bacteria, Candida albicans and the gram positive, E. coli, Staphylococcus aureus, and Streptococci. So we published this work in Journal of Advanced Microbiology Research. So this is one of the peer review index international journal in 2025, volume number 6, issue 1, in page number 128 to 132. So in Google, in this particular journal, you can see our research work. So the title is Showcasing Inhibition Zone of Homeopathic Culture Media in Fungus and Bacterial Culture Assay. So this is the recent published, this is the already a recent advancement in field of microbiology with homeopathy. So this is the integration part that we want. Okay, so this is the recent application. Now next is plant vaccine development. We know about human vaccine, we know about animal vaccine. Now in the plant vaccine, you can see in the figure, this is the antigen candidate. This is the main cause of the disease. Okay, so this is just like a isopathy. Okay, so this is the antigen candidate. When they are derived into two different expression, the stable expression and the transient expression. So when this stable expression, when they introduce in the specific microbe, and when this transient expression introduced in a specific plant variety, so they will give you a specific transgenic plant species. So this transgenic plant species, it may derive from the microbial, the stable expression, or maybe on other hand oblique from the transient expression from any particular virus and the bacteria and the bacilli. When this will give you the extraction of antigen and the edible plants, so this will be going to introduce or going to test in the animal model. So you can see the efficacy of that particular antigen. So here there is a development of serum, blood serum. So these are going to collected, and this is called the vaccine for that particular antigen. Okay, prophylactic measure. So what is prophylaxis? It is just before the onset of disease condition. Okay, so this is the plant vaccine development. Through this process, you can also make the specificity in the recent development in homeopathic pharmacy. So here homeopathy is the plant. Okay, suppose if it is Thuja occidentis, suppose the media through which this extraction of antigen and the edible plants are going to mix is made by the green revolution, which is Thuja mother tincture. So it is one of the method for the recent development in the homeopathic pharmacy. Otherwise, it is just a pharmacy. It is in general. So we want the inclusivity of that particular specificity in the development or the origin of vaccines by plant. So this is one of the important aspect. We can make, this is somewhere it is very complex and somewhere it is very difficult, somewhere it is very heterogeneous. But we want that heterogeneity in their particular mapping, in their particular molecular dimension, heterogeneity in that particular concentration, heterogeneity in that particular constituent, heterogeneity in that particular plant species. Then only we can say it is the hybridization. Then only we can develop the vaccine. Then only we can know the different feature of that particular plant trait. So this is the plant vaccine development. So there are certain method, there are certain procedure through which we can know about this particular procedure. Now bioculture assay. Now this bioculture assay, generally this bioculture assay, it is, suppose this is animal model, okay. This is suppose mice. Now through this we can go for the collection of spleen cell. Now this spleen cell unite with the myeloma cell, which is from different variety, from the different species, from the different group of cell. So one is A, another one is B. When they are going to fuse, so they will give you the hybridomas. So hybridomas are the union of different species and the different varieties and the group of cells A and B. So when this A and B combine, so it forms C, not AB. Again, compounding and combining effect. So here you can see the compounding effect. So compounding is when A combined with B, when it forms C, so it is hybridomas. It is the compounding effect. It is not combining effect. After this hybridomas formation from this spleen cell and the myeloma cell, when they are going to make in a specific culture, in a specific culture media, so you are going to collect the specific monoclonal antibodies from this specific culture medium. So this is the harvested monoclonal antibodies. Now this harvested monoclonal antibodies used as a prophylactic measure. Now here, where is homeopathy? This is allied science. This is the life science. So this culture medium, when we make this medium by the homeopathic concentration, homeopathic medicine, with the specific drug selection, with the specific concentration, specific mother tincture, in a specific dosage, in a specific environment. So this is the homeopathic bioculture assay. Otherwise it is in general bioculture assay. Okay, so here in this part you can make the inclusivity of homeopathy. Next is sources. Now as we know that we have different sources, we have large plant group, we have large vegetable kingdom. Now first, this is the seed treatment. Seed treatment with mutagens. Now when they are going to undergoes into mutations, so they will give you a one specific trait of plant. So this is generation of that particular plant trait called M1 line. So this is one of the trait of plant. When they are going to generate into M2 families, now after this M2 families, when we are going to develop this specific trait of plant species which is different from other species, then we can go for the DNA extraction and the DNA pooling, which we can say the DNA fingerprinting in a normal, in a layman term. So this DNA extraction and the DNA pooling, the DNA fingerprinting is the method of identification of that particular plant trait. It is the method of optimization of that particular morphological feature of that particular plant trait. So this is the DNA extraction and the DNA pooling, which we can say the DNA fingerprinting. In previous slide, we already learned about this DNA molecular marker and the DNA fingerprinting. Okay, so when we go for this PCR, polymerase chain reaction, PCR, so when we go for this PCR amplification with the specific primer, so they will give you the specific enzyme for cell 1. So this is the particular nuclease. Now after this particular nuclease, there are certain mutant identification in that particular loop of, or that particular pole of that particular plant trait. So this is the conformity of that particular mutant individual plant trait. Somewhere it is difficult to understand because it is again not given in any pharmacopoeia, in any volumes of pharmacopoeia, in HPI, in DD Banerjee and in Mandal and Mandal also. But this is a general trait to identifying the specific plant variety on the basis of their specific trait in terms of primer, in terms of specific enzymatic assay, in terms of identifying the mutant in that particular sample, in a given sample. or the seed treatment. After these all are after seed treatment, termination.
Dr.
So
Suresh,
yes, sir.
how much time required? Only 5 minutes remaining.
Just going to finish, sir. Just going to finish in a superficial way, sir.
Okay, we can have the second part
Yes, yes, sir. So
later.
this is about the various method for the sources. Now this is the parameter for plant growth. So again, this is very very important. So how we can collect the drug material, how we can synthesize it, how we can go for the standardization. In figure you can see this is SAM, XRD, FTIR. So these are the various method through which you can optimize the specific quantification of that particular compound which is present in our drug material. So these are the various parameters. Okay, now moving towards the next part. So this is an example of lycopodium extract. So we have alkaloid, we have flavonoids, we have some other terpenoids. So these are the different metabolites which are present in the plant substances. So these are very very important for the optimization. Next is quality and the quantification. So for quality and quantification, we have different parameter like assay of marker, radioactive contamination, foreign matter, foreign substance, microscopic evaluation, macroscopic evaluation, extract value, ash value. So these all are the quality and quantification assessment. So this is very basic. The pharmacopoeia generally relates with the microscopic moisture content, heavy metal detection, total ash value, foreign matter density, microbial and specific gravity. So this is all about the recent advancement. This is the SEM. SEM is the scanning electron microscope. So in next lecture we can discuss about this scanning electron microscope in detail. How we can go for this optimization, quantification and the qualification by the electron microscope for the various drug material. So this will be our next topic. So today our session, our time is going to be finished. So this part is going to conclude it. Next time again I am going to repeat the same slide and I will conclude this part at the final stage. Thank you, sir.
Yes, Dr.
Th-
Suresh Singh. Yes, yes, our moderator is there.
Sorry, sir, because today it is like somewhere in a very deeper sense. But next time we will conclude this work.
Yes, yes, Doctor. It's really a very good session, and you have shown the new ways and new kind of knowledge about this pharmacy, which in the previous uh, era, we never think of in that line. We just simply study the materia medica, what are the things and how it can be used, and what are the medicine prepared from each and everything. And but the scientificity of each and every medicine was entirely different. That's what you have shown. Really great appreciation for your such a wonderful presentation. That's what
Thank
I
you so much, sir.
definitely we can have some more part of the same, second part of the same. Over to Dr. Rijita.
yes, sir. A very congratulations to you, sir. Now,
Thank you.
sir, but I have a question. As you told about plant vaccine development now, sir, in previous Honeyman time, there is rectum injection like this. We have studied in Mandal and Mandal.
Yes, sir. Ma'am, basically, when we talk about this animal vaccine, human vaccine, and the plant vaccine, the most important part is the methodology. So, when we talk
Okay,
about the methodology, so there must be the optimization. Suppose in 11th, in
okay,
11th, 12th, we have one experiment titration. Okay, so in
okay,
titration, there
okay.
is one burette. Okay, and in the bottom part, we have conical flask. Under we have
Okay,
a
sir.
specific
Sir, thank
solvent.
you.
So we
Sir, we are now out of.
want optimization.
Okay, sir. So now we have to forward it to Malayalam team. So very congratulations and thank you. And again, we are welcoming you for the next session.
Okay.
Yes, ma'am. Thank you, ma'am. Next time we will discuss about
Uh,
this scanning electron microscope.
yes.
Yeah.
Tomorrow we will have a session from Dr. Venugopal Reddy, and the subject of discussion will be phobia. That is fear of criticism. that is the subject to be discussed in a different way. So hope all of you will join the same meeting at the same time. Thank you, Dr. Suraj Singh Bhadoria, and to attend the session, and a special thank to our moderator, Dr. Rijita Trivedi. Thank you all.
Thank you.
To Dr. Manoj.
നമസ്കാരം. IFPHന്റെ 2097 ദിവസത്തെ മലയാളം പരിപാടിയിലേക്ക് സ്വാഗതം. ആദ്യമായി ഒരു നിമിഷം മൗന പ്രാർത്ഥന.